Hello, good day. Okay. Howdy, everyone. Today I'm going to uh, present my research work that will be supported by the Xiangzhou Ocean Drilling Fellowship for the next academic year. But before I do that, I would like to thank all the agencies that support me for throughout my PhD journey. First, I would like to thank Fulbright program for supporting me for the first 2 years here at Texas A&M University, and all the lab works that have been done for these preliminary project is from is conducted in our geochemistry and paleoceanography lab at our department, and all the samples from all the samples that I use for these project are from IODP program here. Right. Now let's start it with the presentation. So today I will talk about these beautiful lipids and how we can use them to inform us about the thermal history of our surface ocean across the last 66 million years. It's all started with the first question that my advisor asked me. Did you know that we still don't have a complete record of sea surface temperature for the entire Cenozoic yet? As a person who was not a paleoclimatologist, who was working in an oil and gas industry for a couple of years, I have no clues whether the question is real question or is just oh what eager trying to tease me. Uh, So my first response to the question is that is really because the first temperature proxies that paleoclimatologists have introduced is the oxygen isotopes from calcium carbonate. It was first introduced back in 1950s. So my intuitions told me that it should be someone that should have already completed the Cenozoic sea surface temperature history. So let's guess what I do next after I have been asked this question. So I just Googled it, right? It's what everyone doing these days. Like it's an easy way that we can get an answer to the question we don't know these days. So I searched Cenozoic sea surface temperature, and I found a lot of these beautiful climate history across the last 66 million years. And it doesn't take me too long to realize and ask myself whether eager question is really what he asks or not. So so what are these curves? Let's just get a closer look to these curves. So for those of you who are not in paleoclimate community, I guarantee you that if you ask anyone in the field about what how can you learn about the climate change over the last 66 million years, most of the time you will be directed to this curve, which is the Zarkos curve. And what you see here from this picture is the changes in oxygen isotope of the calcium carbonate from the organisms living in our oceans. And the oxygen isotope of these organisms reflects the changes in oxygen isotope in the world's oceans, which can imply the change in climate or the temperature. So over the last 66 million years, we have changed from a warmer climate, which you can see we have a lighter isotope signals, to the colder climate, the heavier isotope signals, with some abrupt warmer events. As you can see, all of these arrows that pointing to the shorter intervals, but abrupt warmer episodes throughout the Cenozoic. But in general, you can see that like in the early Cenozoic, we have these warmer climate, no continental ice sheet, and we have these temperate forests growing in the Antarctica compared to what we have now, which we call ice hot climates, as indicated by these like black bars showing that we started to have the Antarctic ice sheet developed. And here is the East Antarctic ice sheet with all the cute penguins walking around. So that's the pictures of our Earth climates across the last 66 million years. So you might realize that it's already like clear to us how the Earth's climate changed over the last 66 million years. So why we bothering completing the gaps of these Cenozoic climate curves? It seems like it's no gaps at all from the Sackhouse curve, right? But the problem is that these oxygen isotope data were retrieved from these beautiful benthic forams, these beautiful organisms, where we collected the sediments and we find these calcareous, their shells, the calcium carbonate shells, and their calcium carbonate shells recorded the changes of seawater oxygen isotopes. But as the name of the organism suggests, these benthic forams live in at the bottom of the world's oceans. So when they're living at the bottom of the world's ocean, what their shells recorded was the changes in the oxygen isotope of the bottom water. And according to our current understanding of how the ocean circulations circulate around the world's oceans, we know that our deep water were formed or produced at a certain location at high latitude, right? So for an example, if you're looking at these top panels, it's a cross sections of the Atlantic Oceans. You have these North Atlantic deep water formed in the North Atlantic, and you have these colder, denser deep water masses formed at the Southern Ocean, which is the Antarctic bottom water. So if from this picture, you can see that when you're looking at the Sackhouse curve and you see the changes in oxygen isotope, is reflects the changes at the bottom water. And when we infer it to the surface oceans, it represents only a higher latitude regions. So we still miss a large portion of the surface ocean in the lower latitude that might not be able to see from the Sackhouse curve. So that's when the other proxy comes into play here. So as we know and mentioned already, that our deep ocean, as you can see from this map, the lower panel here is the temperature maps of the water, of the seawater at the depth 4,000 meters, and it's pretty homogeneous. And even though you collected your samples from the lower latitude, like from the tropics, it still shows it's still in the water masses that produce from the higher latitudes. So we still miss a lot of these like low latitude changes across the last 66 million years. So that's why we still need a sea surface temperature record for Cenozoic, because the Sackhouse curve, it's not really inform us about how the low latitude change. So what you see here on the left hand side is what I compiled all the published data of the proxy I will use in my project, which is TEX-66, compared with the benthic delta 018 the Salkos curve that we already discussed about. And first thing that you can see right away is that the number of data between what we have for TEX86, which is our CSFS temperature proxies, is much less comparing to the benthic delta 018. And because, as I mentioned already, oxygen isotopes have been developed for much longer, and people have like collected data for a much longer time. And also they're a global stack, meaning that all of these data are coming from different ocean basins, different latitudes, but it shows a clear trend of how it's changed, and that because of the homogeneous natures of the bottom water. Comparing to these TEX86 data, if we didn't color all the data by the latitudinal zones as you see it right now, you might think that the data is pretty scattered. Not it's not showing any obvious trend comparing to what we see from the benthic delta 018. And that's reflect the wider range of temperature at our ocean surface. So by doing by coloring our data into different latitudinal zones, we started to see some differences here. Like the colder data points represent the higher latitude, and the warmer color data points represent the lower latitudes. And we were talking about it in details more. But I just would like to shows you right away that what we have for the sea surface temperature records across the last 66 million years is not very well established yet. So we need more data for the sea surface temperature records. But why? Like adding a few hundred data points will change the status quo of what we already have or not. Some people might ask, what you're gonna do next when you add it or have it completed. We'll talk a little bit about it. But first, let me introduce you how the TEX86 works as a paleo thermometer, how we can use these lipid biomarkers to inform us about the sea surface temperature in the past. So TEX86 is the temperature proxy based on these organic compounds, as you can see here, which we call GDGT. It's a tetraether lipid, and it's synthesized by the archaea living in the oceans. And the core structure is these long chain carbons. And if you have no internal cyclopentane rings, you don't add, you say GDGT 0, but if you have one cyclopentane rings, you add numbers following the GDGT. So one ring GDGT 1, two ring GDGT 2, and so on and so forth. And there are 6 common GDGT fractions that we generally found in the marine sediments. And we can see the relative abundance of each fraction by doing the LCMS analysis. So on the left-hand side here, you see an examples of the sediments collecting from the colder water and region and the warmer region. If you collected the sediments from the cold regions, you will see that in the sediments, you have relatively higher GDGT 0 comparing to the crenocchio here. And you don't really have a lot of these minor GDGT 1, 2, and 3. Compared to what you have from the warmer region, you have right now much less GDGT 0 and more crenocchio, and you started to have these minor GDGT 1, 2, and 3. And that's how we can tell the difference between the warm and the cold regions. But the TEX86 is the calculation based on all of the minor GDGTs, excluding the GDGT 0 and crenocchio. And by doing these calculation in plotted against the satellite sea surface temperature from the global core top sediments, you can see that these sediments, the Tex86 values from these core top sediments from all over the world shows a strong positive relationship with the sea surface temperature. And that's how we can use these Tex86 values from the marine sediments as our thermometers. For an example, if you have a Tex86 values of about 0.5, it reflects a sea surface temperature ranging from like 15 degrees Celsius to 20 degrees Celsius and something like that. And let's talk a little bit about why I choose Tex86 over other temperature proxies. Even though there are like a lot of temperature proxies available in the paleoclimate community, but Tex86, it's good for reconstructing the sea surface temperature in the very old geologic times intervals. It's less impacted by the diagenesis after depositions. It also shows good relationship between the Tex86 values and sea surface temperature, as you see from the previous slide. It has been used in several studies that reconstructing the temperature in much older time interval, like than what I'm doing right now, and comparing to the time using for collecting the data, it's much less time consuming comparing with the planktonic forams that will have to find the very well preserved shells to do a good oxygen isotope work. But there are some cons about the proxy as well, because it's an organic compound. So if we collected it from the very matured or over mature rocks, it can have very low abundance of these organic compounds because it's already expelled into hydrocarbons. And because it's relatively new proxies, we don't really understand we still have a lot of question about the biology, about who produced the GDGTs and stuff like that. But in a simple terms, we can still use it as a sea surface temperature proxies. ขอหยุดก่อนได้ไหม เดี๋ยวรู้สึกว่าพูดเยอะเกินไป Now, after you know how to use Tex86 to inform us about the sea surface temperature in the past, let's just take a closer look to what we have in the Tex86 records. As I mentioned already, our main mission is to produce the continuous Cenozoic sea surface temperature records. So I'm not going to like add a couple hundred data points anywhere. I have to target it to a very specific locations and a very specific time interval. So here, the Tex86 data are coming from like 75 drilling sites over 1,900 published data. And most of the studies that publish these Tex86 data are focusing in this very specific time interval during the Cenozoic. So what I'm going to do for my project is trying to fill these geologic time intervals that have low density of data points. And to do that, I have to find sites that have a potential to produce the Tex86 values to fill these gaps. All right, so let's just do it. Like let's see how I can do it. So my samples that I will use for Tex86 coming from deep sea sediments, and all of the samples were drilled and cored by the IODP program, which have 3 main core repositories located around the world. And we have one hosted here at our West Campus, which is the Gulf Coast repositories. And there are over thousands of the sediment cores that have been drilled since 1966. So what I have to do is to find sites that have a virtually complete sedimentary sequences that deposited continuously across the last 66 million years. And also having a relatively high sedimentation rates, because high sedimentation rates will at least giving you a higher chance of enough organic materials that we can use to extract a measurable GDGTs. So these are my potential sites that I first selected to perform my preliminary uh, experiments. And here is how we do it. We collected the sediments from the sediment cores at IODP core repositories, and then we freeze dried them, we homogenize them, we ground them with these pestle and mortar, and we extracted the total lipids, as you can see here. Like there are like different sediments have different organic or lipid compounds and having these different colors. Then we purified the lipid fractions for the fraction that we were interested in, the fraction that we aim for finding the GDGTs, and then we run all the purified fractions through these LCMS or the lipid chromatography mass spectrometry machine. And here is the preliminary results of all the samples that I collected from all the preliminary, all the potential sites. So again, you're looking at the TEX86 records from all the colored data points are the published data, and coming from site that you can see from these map. And the color of the data point corresponding to the latitudinal zones that you can see from the map as well. And all the black data points is my data from the sites that I plot here as a red dot colors. So I would just point it out some examples of the sites. For an example, these circle data point here, it's aligned well with the light green and dark green color published data, right? And these data point are coming from site 1172, which is a little bit south of Australia. And it's from the modern location, it's located in these light green regions, which is the southern hemisphere mid latitudes. It's showing that our preliminary results is aligned with what other people have been published. And these site 1172 have giving us promising results in terms of having the TEX86 values that in in the interval that people haven't really published the TEX86 yet. So this is how we're gonna fill the gaps of these TEX86 records. Like these sites, it's have a potential to fill the older interval and also like the younger interval in the mid latitudes. Similarly, what we have in the tropics or low latitudes, like for an example, the data from site 929 here and site 807 here, it's 929 is coming from tropical Atlantic and 807 is coming from the Western Pacific warm pool, and it's also show an alignment with what the published data shows us from the tropic regions, which relatively higher TEX86 values comparing to the higher latitude region and stuff like that. So 807, 929, and 1172 is our three main sites that we will use to fill in the gaps of these TEX86 records, right? And that's how we're gonna do it. Now, after we have, like hopefully if we have a complete records of TEX86, what we can do further other than only filling the gaps. We can just, first we can just comparing with the text with the benthic delta 018 data and see how is the relationship between the two proxies, right? And it might inform us about the relationship between the bottom water and the surface oceans. So to do that, we have to smooth the data, because benthic delta 018 were collected with different location, different time steps comparing to the text86 data. So we have to do these running mean curves for the text86 data and the benthic delta 018 in order to compare it at the same time step. And let's see how it's looked like. But before I show you the actual results, I'm just going to briefly point, like guiding you what we expected from these scatter plot. So what you will see is the comparison between the oxygen isotope of benthic 018 versus the text86 values. And right now, the heavier oxygen isotope is going to the left-hand side and the lighter isotope is going to the right-hand side. And if you see a steeper slope from the plot, it's meaning that what changes at the surface is very co-vary with what changes at the bottom oceans. But if you see a little bit flatter curve, a flatter relationship, it's mean that it's what changes in the bottom ocean is not really corresponding with what changes at the surface ocean and stuff like that. And here is the results. So the color of the data point is corresponding to the latitude, zones, the same as what you see from the previous slide. The warmer color coming from the tropical zones and the colder color coming from mid to high latitudes. And the green data point is from the southern hemisphere and the blue data point is coming from the northern hemispheres. So the first thing that you can see right away is that there are two distinct trends here. The warmer data points show a flatter slopes comparing to the green data points. And it's mean that the low latitude, it's acting different from the high latitude surface oceans. And what it can really mean is that across our last 66 million years, what we see changes at the bottom ocean, it's highly correlated with what changes at the southern hemisphere, mid to high latitudes, which is aligned with our bottom water in the modern oceans. Our majority of the water at the bottom of the ocean is Antarctic bottom water. So this is not out of our expectations. So it's inform us what we should see. So that's a good sign. But you might see these like odd blue data point that show the opposite trend compared to other regions. And that is coming from Antarctic oceans. And that's not out of our expectation as well, because for text86 proxy, we most of the time excluding these Arctic sediments because of the more complexities, different archeo community, and like a lot of question that we still don't really understand how the text86 works in the Arctic. So it's meaning that these opposite trend just confirming us that the Arctic oceans is a different monster since the last 66 million years. So that's how we can use the text86 data to inform us about the more dynamic pictures of the surface ocean beyond what we see from the benthic delta 018. So what I'm going to do next is just as mentioned already, extracted more samples in the targeted geologic time intervals from the potential sites. And after we have the complete records of the Text 86, we can do something more, a little bit more fancy, like we can see how the meridional seafloor temperature gradients change over the last 66 million years. And we can investigate the relationship of the surface ocean with the bottom ocean, whether or not the ocean circulation has changed or it remains the same across the last 66 million years. So then I just would like to end it my presentation by saying that I hope I shows you that what we have now as the global stack benthic foraminifer delta 018 only reflects a portion of surface oceans. It's captured only high latitude, and we still have not really established how the low latitude surface ocean changed across the last 66 million years. And also there are two potential latitudinal zones, which is the tropics and the southern hemisphere mid to high latitude. So we can fill in the gaps using text 86 values, the new text 86 data that we will produce from this work. And the preliminary investigations of how we can use text 86 to inform our Cenozoic thermal history beyond the benthic delta 018, giving us a promising result that we will find something new about the Cenozoic thermal history beyond the Saar-Korsberg. And that is our prime ultimate goals. And that's it. All right. จบแบบงง